I am delighted to be able to introduce friend and colleague Jonathan Dursi, who's coming from the Center for Computational Medicine in the Hospital for Sick Children in Toronto. He started his academic career as an undergrad at St. Mary's University in Halifax. He did a master's in physics at Queen's. He started out doing lattice QCD and then wound up going into galaxy simulations, which, as he put it, was stepping up a few scales. He had a year at Waterloo that he doesn't speak about, and then transferred to the University of Chicago, where he was in the Flash Center with Mike and me. He was there on a Krell Computational Science Graduate Fellowship, and the three of us, while we were there, were inducted in the Joseph Sheffield Memorial Society. Following that, he did a postdoc at CETA, and then at the SciNet High Performance Computing Center, where he was on the staff. He then went to the Ontario Institute for Cancer Research, and as I said, is now at the Hospital for Sick Children, where he's going to tell us about HPC, big data, and large-scale genomics. Dinner is going to be at the bench, and if folks want to join, we're going to converge there around 6:30 or so. Please feel free to come along.
Thanks, Alan. So I love coming to places like this because my interest is in computational science pretty broadly, as you may have guessed from Alan's recitation of my checkered past. But it's a tough crowd to give a talk to, right? Because everyone has deep computational backgrounds, but in very different areas. So like a deep dive into the genomic stuff I'm doing isn't going to be interesting. So I'm going to try doing something a little different this time. So I'm going to start talking with the high-level stuff that I've been sort of worrying about / sort of doing something about, and talk about why I think that matters. And then towards the end, sort of talk a little bit about the sort of things I've been doing and why these plug into what I'm been thinking about. And so what I've really come here today to tell you is that there's a lot of talk about the convergence of HPC and big data. And if this is actually going to be a thing that's actually going to happen in the near term, as opposed into some dystopian exascale future, it's going to be because of the needs of large-scale genomics. And if it does happen for large-scale genomics, it's going to happen because of places like this, where there's a bunch of people with very different expertise in very different areas who can actually all talk to each other and get some cool things done. So I've been working at this particular Coalface for a little while now. Alan's already told you I was at the DOE ASCII Flash Center. I was doing real hardy, stick-to-your-ribs type HPC, Fortran, MPI, adaptive meshes. Joined an HPC center after a postdoc, doing a broader range of science, a slightly broader range of tools. And it was really, really cool to see all the different sorts of problems people had. And this is so at the start of my career, things were very stable. There'd been this 20 years of kind of things being the way they were. Big computing was MPI over a whackload of x86. No one outside of academia was doing much of this sort of thing. There was a pretty stable set of problems. This is my picture of the state of the world in this more innocent time. It was a simpler time. There was statistical computing was largely the realm of social scientists, a little bit of experimental science. Physical scientists did the sort of thing that we were just describing. Not a lot of sort of database-y stuff in academia, but it's a little ferry that ran along this river between the statistical computing side and the sort of enterprise computing side, and maybe very infrequent ferry service between the statistical side and the MATLAB side. And then they came. So the widespread adoption of computing and networking brought a ton of data really, really quickly. The first people off the bat were people who stood to make money off it, these internet-scale companies. And so they generated things like Hadoop and HDFS, which just generated an entire ecosystem. And just sort of by dumb luck, genomics was just becoming a thing at the same time. Right place, right time. The Human Genome Project had just sort of wound up successfully in 2003. High throughput sequencing was starting to become a big thing. There was lots and lots of data and how to process it. And crucially, these sorts of problems were arriving on the scene after HPC was already quite mature and quite optimized for the problems it was solving. On the other hand, big data was just this wide open thing. And I do want to remind everyone how new this is. I've already been asked once about, like, can a physical scientist meaningfully transition into something like bioinformatics? Bioinformatics is like 10 years old. Nobody knows how to do bioinformatics now. It is very, the field is wide open. So I started going to genomics in 2013. They have huge computing needs, super interesting algorithmic problems, and hey, HPC to the rescue. I got this. So I made the move in 2014, Ontario Institute for Computational Research, working with Jared Simpson, who amongst other deeds was the author of Abyss, which was one of the first open source genome assemblers that could actually work on human-sized genomes. And to do that, it was distributed memory. It was MPI-based. So I'd clearly found my place. Abyss 2.0 just came out. Didn't someone maintaining it after us, but it came out with the new default mode being non-MPI. And the phrase "never again" came out. In the meantime, GATK, another basically one of the canonical genomics toolkits, now finally supports distributed memory computing with Apache Spark, not with MPI. So this is what I think the world looks like sort of today. Right now, genomics is sort of wedged in pretty close to big data expanse, but it's not thriving there as well as it could. So I'd claim that if anything is going to bridge these two solitudes, it's going to be genomics. It's going to be genomics because it's fundamentally data, not simulation intensive, truly large amounts of data, super irregular. The problems on this data look more like NoSQL databases or social networks than they do like PDEs on grids. But it's fundamentally science. It's fundamentally doing really subtle analyses on this data. Not just subtle analyses, but asking questions that have never been asked before on this data. And amongst other things, this means that testing and discovery of methods means that your methods really need to scale down super well.
What do you mean by scale down?
So I mean the following. So anything will scale if you throw enough data at it, right?
Sure.
But that's not how scientists work, right? When we're trying to find out a problem, when we're trying to figure out, okay, first, is this even a question that's sensible to ask? Two, if it is this a method which can be used to ask this question? You can't do that on terabytes or petabytes of data. It just doesn't, you're just gonna be lost in the weeds forever there. You need it to kind of work on a laptop or a workstation or a couple nodes, right? And none of the big data sort of frameworks do that. They're not designed for that. Okay. And this is one of the big reasons I think why Spark, which has other problems for science, but one of the reasons it's just not a physicist-friendly tool, because you can, in principle, run Spark on your laptop, but it's useless. Right? It's just such a heavyweight framework, and that's fine if your use case for it is petabytes of data. But the beauty of HPC tools is that they typically can be meaningfully tested on kind of a laptop workstation sort of thing. And this is a feature that is utterly lacking in most, not all, of the sort of big data frameworks.
Why did it evolve that way? I mean, if you look at this sort of simulation stuff that we do, that you used to do, it doesn't always range from,
Right.
laptop scale to...
Yeah. So why did it evolve that way? I think because, and maybe this is changing a little bit with things like TensorFlow and deep learning or whatever, but I think up until this point, the questions that we're asking were pretty well understood, right? You were going to do a clustering algorithm. You were going to use one of 5 that exist in the literature. You could, if you wanted to test it, you could do it in Python on your desktop or something, then just immediately splat it to Spark as a framework. But I think for real sort of science, as opposed to sort of a production thing, like once you have a method you know that works, you can build some production thing and invest whatever time and energy you need to, because it's going to be run 10 million times. So you can kind of do that for science, but when you're just getting started, you're asking deeper questions than most of the big data stuff is doing now. I think this is less true with deep learning stuff, where they're still sort of in that same mode. They're trying to figure out, well, does this whatever convolutional neural network make sense in this case? There, you're starting to see that, and so TensorFlow will run on your laptop, but Spark just won't. It's a big statistics package, mainly implementing fairly well understood tools. So that would be my argument. So eventually, I claim these two blobs are going to end up touching each other, but I think genomics is making this bridge faster than anything else, and it comes out of immediate needs. For HPC, they recognize that it would be good to have some of these big data-like approaches, but it's focused on exascale, which is fine, but it's more future-looking than genomics needs. Big data is sort of trying to relearn these HPC lessons as needed, but it's doing it one bit at a time. And so you have individual projects which are maddeningly have a piece of the puzzle that genomics would want, but not all of them. So yeah, it's going to take a lot of people with a lot of different kinds of knowledge to make this happen, to help with genomics research now, and to help transfer expertise between big data and HPC, so they're not each relearning the same thing. And it takes places like this to bring these people together.
What if we build a wall?
And make them pay for it.
Which them?
All of them.
Big data.
So I'm going to start with why I think this convergence is inevitable, why I think genomics is a meaningful driver. A couple of, won't say increasingly, but a number of hybrid genomics projects, or projects that have a little bit of each, and a couple of things that I'm excited about in the future. All right, so I claim this is inevitable because the two camps started in different places, and they're on different paths. Simulations, as scale goes up, are getting more complex, more dynamic. They just have to, as resolution goes up, as more physics is involved. And big data problems, they have amounts of data that are going up, but that's not the driver. They want to ask deeper and deeper questions of the data sets they have. And all the while, the underlying mathematical tools you have available to you are the same. So HPC's path over the last 20 years has largely been about scale. From this ancient machine, scaling up, the story's mainly been about hardware. And the fundamental software stack has remained largely unchanged, although the adoption of things like OpenMP, OpenACC, OpenUH, things like directive-based programming for accelerators does show that there's willingness to adopt new approaches when they're compelling them. Big data's path, like I say, is more, takes a volume of data, it's more or less a given, and its scaling is less about physical scale and more about extracting more information from the data, asking deeper and deeper questions. Started with MapReduce, which was, PageRank was sort of the height of sophistication for what you could really pull off with that. Start moving to streaming data, start moving to, actual machine learning, actually fitting fairly complex models of some sort. And so eventually those things have to hit each other, right? Because whatever you do with simulations or data, eventually you're going to have to solve a linear algebra problem. And in big data world, it's very unstructured sparsity. And as you go to larger and larger scale on for something like a PDEs, your sparsity patterns get less and less structured too. You just start being constrained by other things than the geometry, and the sparsity patterns start getting weirder. You see the same thing in sort of graph problems. Social network graphs are maybe not power law distributed, but they very irregular. Nodes can have very varying degrees. Inexplicably, even though we're Canadian, and my tweets on HPC are extremely apropos. Justin Bieber has many more followers than I do. These are the sorts of graphs you'd like to be able to do in big data, but when you actually do something on these complex graphs, you actually do exactly the same thing you do in an unstructured mesh. You do some neighbor exchanges, do some calculations. It's not a super weird thing. And in fact, if anything, these social network calculations are more latency sensitive than they are in HPC because the calculations are lighter. And of course, time series. I mean, a time series is time series. You're going to be doing FFTs. You're going to be doing some correlation analysis. There's only so much math available to you. So these problems end up being the same, and these two groups are going to collide into each other. On the HPC side, the main deliverable of this has been white papers and vendor press releases. But there's starting to be some cool stuff out there, which I'm actually really pumped about and would be more pumped about if it wasn't something aimed for 5-10 years from now. So data flow is starting to become a common way of expressing complex calculations on both the big data side and the HPC side. You set up the flow of data and you let your compiler and runtime, maybe with hints from you, actually decide how that data gets distributed and what the execution plan is. This is, when it works, it's a tool for abstracting away the architecture, trying to get some performance portability. This is not a crazy thing to do. SQL databases have been doing this sort of execution planning and optimizing steps for a very long time. The trick is doing it in a distributed way. Also on the HPC side, there's starting to be these extended PGAS languages with very deep runtime stacks to try to do some of this reordering at different levels. Again, super cool, but genomics would like something sooner. HPC, or big data, really just sort of thinks of improving performance. It doesn't really think about trying to converge with HPC. And there's a few things going on the big data side. There's databases where the core engines are starting to look very HPC-y in terms of extracting every little bit of performance out of a node. And on the machine learning side, they're starting to have more coupled iterative computing. It's leaning more on software design than on hardware. And again, infuriatingly, they're different projects. ScyllaDB, this very HPC-like, extract every little bit out of commodity hardware with C++ and every little network trick you can imagine. Farm from Microsoft Research uses RDMA and some remote procedure calls over SSDs to get just absolutely insane, like very HPC-like latencies. These are, when the network is practically flooded with requests, this is making an arbitrary distributed database, distributed dictionary lookup over a couple terabytes of data on disk, on SSDs, over 20 machines in 30 microseconds. I mean, this is starting to be performance you don't necessarily scoff at. This is actually pretty good. So on the database side, if all we wanted to do in genomics is query a database, and we were done, there'd be tools for that. But there's more than that. We want to be able to make these queries, but actually integrate them into calculation. There's tools like Spark, which are getting more and more performant, which is great. And you can build things on, but it's still fairly slow. There's things like TensorFlow, which are building sort of, again, a data flow execution over tensors. It drives me nuts. They're n-dimensional arrays. They're not tensors.
Yeah,
Right? Right?
It's crazy.
I'm going to write a letter. So you know, this is starting to look more HPC, but it's super, right, that you're actually doing real calculations, threaded GPUs on n-dimensional arrays. That's starting to look better, but it's all so primitive. You can, legally mandatory Mandelbrot example, little PDEs. But you have to distribute a computation. You actually write the chunks of a computation out task by task. So both of these sides are sort of groping towards some of the same solutions. Both sides are sort of capable in some cases of taking full advantage of hardware. Data flows and models starting to be useful. Higher level languages are starting to take place here and there, but no one tool set has all the pieces. And this is what's sort of frustrating to us in genomics. So I tend to think of genomics as having two broad sort of types of workloads. So one is very search heavy, indices, graphs, databases, sort of typified by genome assembly. Has everyone here seen a talk about genome assembly and bioinformatics?
Everyone here is assembled from genes.
So the idea is, you take a bit of person, turn it into little segments of genomes, maybe 250 is sort of the standard, 250 base pairs out of 3 billion. And you try pasting these together. And you have to look up, if you have some chunk here, GGA, does the potential neighbor GGA something exist? And so you need to do these sorts of operations. Assembly isn't actually something you do necessarily super often, but most of this sort of workflow happens a lot. You're trying to do string indices or string databases somehow, and one way or another build some implicit graph and traverse it. It's always much messier than pretty little diagrams. You always end up getting fragments. This is a mosquito genome, the best available, but it's broken into like 300, 500
Do you
pieces.
really use strings? I mean, there's only 4 letters.
So they'll use strings, but it'll be 4-character alphabets. Though sometimes more than 4 for base. This is so. Do I have a better? Here, let me skip it. Ooh, I may not be able to find it. These are pieces of a graph. So these would be very small nodes in this graph. This is one of those extended a great deal, but assembly, especially with these short reads, you can't resolve repeats. Like if there's a cycle, you have no idea how many times that cycle actually executes. And also, just by bad luck sampling or noise, you will get fragments. You have this, but you don't have this or this, so you can't connect it to it. So a real assembly, if with just short reads, ends up being sort of a fragmented mess, and you have to use much more manual approaches to sort of map on where an actual leaving the realm of computation, and now you're actually you're actually talking people in a lab trying to figure out where would that actually go.
And things like the jumping gene make that even more complicated.
Exactly. Things are a little better. I'll talk about nanopore reads. There's long read technologies which are starting to help stitch these together.
I like a team of rival scientists.
Yes. So, and sort of there's that, and there's sort of more traditional statistical stuff, but the stats problems get really big. So instead of ripping my genome up into a bajillion little pieces, 100 million. So now let's say we're leaving just DNA and seeing the RNA that it gets transcribed to, sometimes on the way to protein. So take that from one of my cells, the little RNAs, and sequence that. Figure out which genes are being expressed. But now do that for the different types of cells everywhere in my body, right? They all have the same genome, but they're the same DNA, but they're expressing RNA very differently. And now do that for a bunch of cells in my body, maybe in a sick or a healthy state, and then do that across a population and start correlating it with other things. So you get these truly large genomics sort of statistical problems, which are cool, but they're a bit more structured. Okay, so those are the sorts of things people try to do in genomics. So I think anyone who's ever worked in an HPC type center has asked themselves this question, and this is sort of result. These tools are not made for these problems, which is fine. These tools are made for more regular data access. There's an OMP parallel 4 doesn't make any sense in like a database typically. That's not the sort of thing you have. These sort of neighbor operations, they can, but it's a little more complicated. Distributed databases don't typically use libraries that look like MPI or OpenMP. People do in fact try it, and they succeed, and nobody dies from it. But it's really, really hard, and it's really, really hard at a time when people are still developing the method. So going back and adding a new assembly method to one of these codes is super, super tough. On the other hand, people do have somewhat more luck with PCAS tools, so it's not. On the other hand, okay, so why don't the genomics tools just use these databases I talked about that had really good performance? Well, this is, again, sort of C figure 1. It just doesn't work out so well. There's these very performant, low-level pieces, but they're databases. They're not like libraries. There's no meaningful way to compose them into something meaningful.
What about like the task-based frameworks?
Like what?
Coalface or something.
Yeah, so I think that's actually starting to look more useful, I think. And I think that sort of ties into a little bit of, I guess maybe because I learned about them at the same sort of time, I tend to put them in this sort of execution sort of the data flow model, which isn't really right, but I think those do have some promise, but they're just so new. And on the other hand, using these tools like Spark isn't any better. They're too slow, and this is what we were saying earlier, especially at small scale. There's this actually scathing paper, which if you enjoy reading papers that tear apart other papers, I can highly agree to you. Highly recommend. It's easy to scale when you have really, really big overheads that you gradually overcome with more and more data. This is a paper just exploring the scalability of some of these tools. That are these things that are shown in the literature to scale very well, but comparing them to a single-threaded execution for a single thread running on a single node for some of the same benchmark problems they publish, and they just get absolutely slaughtered by well-written single-thread codes for these small problems. Scaling at really large scale isn't necessarily that hard. Scaling on small scale turns out to actually be super subtle.
So what is the problem? I mean, what changes at small scale?
So Spark is a good example. Let's take Spark as an example. It's lovely, has very nice architecture. But when you're building something for petabytes of scale, you don't mind a ton of like startup time. So I mean, for one thing, just these JVMs turning on is already just crazy when you're talking about dozens of seconds. But they have all this infrastructure for distributing computation and right at the beginning, and it's just super heavyweight. And it's not even that it's bad. It's not appropriate for these size problems. Like if your data set isn't filling up dozens and dozens of disks, you have no business firing this far.
And yet people do it. So this infuriates me. So I'm going to share it with you. So the Broad Institute in Boston put together this Hail project, and it's a tool for a user, a researcher, asking questions and doing aggregations. Sort of, does person X have genetic variant Y in them? It's a two-dimensional array. It's you're interactively doing this data exploration. It's all good. It's all fine. There's nothing wrong with it. But it's in Spark because it's all they could manage to find to use to tackle this problem interactively. And a big problem in this case is like 1 billion entries, and each of these entries has, I don't know, a dozen numbers. This is not a big problem, right? It's very unwieldy for researchers on smaller sets. And I claim that just the fact that this even exists is a failure, is a mission of failure by some of us who work at HPC centers or whatever at providing decent parallel computing tools for sort of genomics problems. Right now, doing large genomics means going out and buying huge RAM machines. So the good news is there's tons of amazing work being done, including by people here, on these sorts of problems, succinct data structures, sketching and streaming methods that are approximate or even exact. There's fantastic work, but they're doing it because they have to, because there's no meaningful way to do distributed memory problems in some sort of fairly flexible way.
Jonathan, a completely tangential question.
Yeah.
What about the privacy implications of these data sets?
I am super glad you asked that. And I would be happy to answer that question in 15 seconds.
Okay.
I think
Is
so.
it
That's
about the Cambridge Analytics thing? Or?
that's right. I knew you would ask that question, Mike, because I've trolled your troublemaking social media history. So genomics data is doubling every 7 to 12 months or whatever. Again, this is a traditional slide to show, and they're enforcing it now in all genomics talks. So here it is. Genomic volumes are doubling at every 7 months. And my usual sort of sunny side, I claim this underestimates how terrible it's all going to be. So there's new devices arriving, which are going to move some sequencing from specialized core facilities to small labs. So some of us have seen this movie before. Instead of having to ship up, send your data to some center that does this, and 3 weeks later you'll get it, biologists are going to be able to do it on their laptop. Entirely new data types are becoming available, like direct RNA sequencing. And all of these problems, changes in research genomics, are going to be small potatoes compared to the actual real elephant in the room, which is genomic medicine. So whole genome sequencing is already starting to become part of the toolkit for rare diseases or some oncology cases. $1,000 for a whole genome or whatever is a lot for a test, but it's not unfathomable. And as this becomes cheaper and faster and easier, it's going to become part of the standard of care for some treatments. And medicine is absolutely enormously mind-bogglingly huge. Like in the US, just hospital spending is 10 times the entire NIH budget. When medicine starts using genomic data routinely, it's going to swamp any research use. So if this were just about doing the larger number of the same sorts of analysis, that wouldn't be so bad, right? You just need more of the same sorts of resources as we have. But it's actually worse than that. The more data and the better techniques make possible, in fact necessary, different kinds of analysis that involve combining lots of different, looking at lots of data at once. So metagenomics is one case where here's another one of these graphs. It's a similar sort of graph, but in this case, each of these things is legitimately a different critter. So you take a sample of stuff with microbes in it. With human health, it could be wounds looking for infection. It could be from the environment. It could be looking for pathogens. It could be environmental. It could be gut flora. And you're trying to figure out just from these pieces of genomes what all critters you have without even knowing how many critters you have. And this is already complicated, but now imagine starting to do this with a time series, or try to imagine having a data set of every known sort of microbe you might want to probe for. These start getting really absurdly huge. On the population genomics side, right now on human health, we tend to think of things in terms of a single reference human genome, which is just an absurd concept. There's huge amounts of variation and diversity in people, and especially in some genomic regions. There's anything to do with the immune system. It's wildly variable. And there's known differences between populations, and you get founder effects where you have certain variants that just stick with that population, tend to get fixated in that population. So rather than having a single human reference, it makes more sense to build a graph reference that contains large amounts of the variants, But now creating these graphs is non-trivial, and using them is much more complicated and requires much more memory than a linear reference. And we've talked about how quickly genomic RNA sequencing goes crazy. So let me tell you about some projects. So the approach in genomics has been basically to try to pick and choose bits of technology. So there's excellent tools that make full use of a single node, and often you're trying to integrate. You're trying to run this very complex pipeline of tools using sort of cloud or big data tools. So I'm going to talk about 3 projects, how they're straddling this, and what would be possible if the divide were narrower. And I was not careful enough in checking my slides. My GitHub page has not updated, so it doesn't have some slides. So I'm going to talk to you for the rest of this project, or for the rest of our time together without the slides. So I want to tell you about 3 projects that take different pieces of this. So one of the projects is nanopore sequencing. And I'm going to go back and show you that nanopore slide. So this is a straight out this is a straight out,
numerical computing problem. Genomics data types are getting, much richer. They're much more complicated than they used to be, which is awesome. So we have this little device here, stapler sized, and DNA gets fed into it with a little protein that's much like what happens when it's actually transcribing DNA in your blood, in your cells, and it feeds it through a little pore, and this entire thing is filled with an ionic fluid. Current goes through, and as one of these bases works its way through here, it interacts with the pore, and the amount of current that's going through changes. So we have a straight out signal processing with actual, honest to God, floating point numbers coming from a sequencing experiment. And so now we have to calculate. So this ends up looking like an absolutely bog standard sort of HPC thing. You end up with using GPUs. You end up sort of having loops for logarithms. We got lookup tables for logarithms for likelihood calculations. And so you can distribute these sorts of data sets. But again, you need to train these models. And this training ends up looking much more like a big data problem. You have a large amounts of data, you orchestrate the running of these things. Another problem I want to tell you about is the pan-cancer analysis of whole genomes, which is a project that's just about to come out, or the papers will be announced tomorrow, where we needed to analyze, it was a huge international collaboration involving about 20 countries, about 30 cancer types, sequencing data from all over the world. Some of that data could not leave their home countries. And we needed to run an analysis pipeline, a uniform analysis pipeline, in all 2,700 of these paired tumor normal samples. And we needed to do it in such a way that people could actually make biological inferences at the end, that people could take a look at this data and say with some certainty, holy cow, looks like these variants are actually associated with these forms of cancers and not these others. So this, again, you have specific individual tools that look like HPC, but trying to orchestrate this over an international consortium, over a bunch of different clouds and HPC clusters. These are not, these are tools that the big data, the cloud side has, and works really well. And so we need to adopt those. So we were very early adopters of Docker. And there's this store, docstore.org, where you can actually go and get all the Dockers from for each of the tools, and the common workflow language, the orchestration language, allows these things to run. And when you're trying to deploy updates across all these different clouds, and you're trying to make sure everyone is running exactly the same pipeline, this is the only way to do these sorts of things. These tools exist in the big data and cloud world, and they're just starting to make appearances in HPC world. And that's great. That's an example of data transfer coming over. But what really gets into this convergence is the problem I'm currently working on, which is CANDID, the distributed infrastructure for genomics. So in Canada, health data is a provincial jurisdiction. And it's a very big deal to get health data, to ship it en masse between provinces. They take the privacy extremely seriously, and the privacy regulations are different between provinces. For research data, it's not such a big deal, because when you're putting together a research project and when you're consenting data for a research project, which is a very labor-intensive process, you can write the data sharing agreements as part of this. For that particular research project, these particular researchers can access the data. But by 2020, 2025, almost, the
majority of genomics data is going to be health related. And you simply can't negotiate these arrangements for every single person. You wouldn't want to. It's a crazy idea. You wouldn't want health data to be routinely visible all across the country. So we're building in a distributed infrastructure where each hospital, each data steward, keeps its own data and provides programmatic analysis of the data. You can query the data with a list of analysis, and the sites can send their data back, making sure that not too much information is leaked. So we're building up this infrastructure, and the stack doesn't look anything like HPC or even big data. It's all Go and web services. And it might seem completely sort of irrelevant to this conversation, except the privacy implications are getting more and more finer grained. So in this post-Facebook scandal world, and in North America and Europe, and those patients are getting more and more ownership of their health data, and are starting to become, in some places, allowed to determine who gets to see it and how much access they have. So now this sort of computation, performing analysis, a distributed analysis of data through this exchange of messages over web interfaces, as more and more people get control of the privacy of their data, which is a good thing, now that data may not be, it's not a question of exchanging messages between a hospital in Nova Scotia and a hospital in British Columbia. It might be actually two nodes in the same rack in some cloud data center. And the issue isn't that the data couldn't be in memory because of technical reasons. The issue is that the data can't be in the same bit of memory because of privacy regulation. So now what we have is a bunch of nodes in a rack, each with different views of a larger data set, that now are exchanging information via messages with each other to create some distributed computation. So this is now actually an HPC problem. We are actually doing distributed computations with messages in a rack or in a data center or across the country, and we need to be able to do that efficiently. Except the messages aren't, here's my guard cells. The messages are the DNA equivalent of go fish. Do you have any variants that are this? So what we are going to is very much HPC plus big data.
How do you know some of that can't craft a query to reconstruct the data set?
So there's a big literature on that, and to actually totally rule that out would probably be so restrictive you couldn't do anything with it. But there are really interesting things you can. So the first way you avoid this is you only have a prescribed list of queries you can ask. So that's how you start the problem. You do what you can with those. But there's a bunch of interesting things in differential privacy, which we can talk about a little bit too, which means you can't guarantee that some information won't leak, but you can put error bound, you can put probability bounds on that, and that starts to seem like a fairly reasonable way.
But who sets the probability?
So that has to be some regulator, or it could be actually the user, right? If it's going to help research this rare disease that my son has, set a 10% error bound that you can find out who I am or something. So to solve the problems I want to solve, I want there to be tools that have big data type flexibility to actually perform like HPC code. And there are things that I'm really genuinely, uncharacteristically optimistic about that can make that happen. So, and a lot of them are the sorts of things that are happening by people affiliated with ISAs. with the introduction of non-volatile memory, external memory algorithms start becoming extremely exciting again. And with non-volatile memory plus RDMA on converged Ethernet networks, you can start being able to do distributed external memory algorithms. So you start being able to do these very complicated string databases, but in a fast-performing way. With improved PGAS languages, like the PGAS language, Shmem and its successors like OpenShmem, but also with things like Chapel that have built-in distributed associative arrays. That starts to look promising. With these new execution data flow type models, including these task-based models, there starts to be promising. And with the genomics work and with the algorithms work that's going on at Stony Brook, it's really exciting. This is probably the most exciting time in biological science probably there's ever been. But the only way we can routinely get answers to the new questions we're thinking of asking is having centers like this that bring people with very different expertise and interests together so they can talk to each other. So this, the fact that being invited to talk at IACS is actually super exciting, because this is the sort of place where stuff like that happens. And that's it. Thanks.